Hello, everyone. Thanks for joining. And today, uh, we will be talking about CRISPR-Cas9 system in treating diseases and beyond. Okay, I actually don't usually put memes in my presentations, but this was exactly the one I was looking for. So when we talk about CRISPR-Cas9 system, we should first of all think about bacteria and be thankful for them because this system is naturally present in bacteria, and bacteria use the system to defend themselves against viral attacks. Now, how do we do that? So viruses have PAM sequence that is recognized by bacterial guide RNA. So that guide RNA is complementary to the viral RNA. And after these two sequences come close to each other, the Cas9 enzyme binds to the guide RNA, and then this enzyme cuts the target sequence in the virus. So this enzyme uses RNA molecules that guide RNA to specify double-stranded DNA sequences for site-specific cleavage. And the important CRISPR region is composed of Cas nuclease, track RNA, and cRNA, and the two RNA are complementary to each other and make up guide RNA. So as I said, that guide RNA guides Cas. So here you see binding guide RNA binding to the DNA in which target sequence is present, and then Cas9 binds there. And Cas9 is called also a nuclease because it coats nucleic acids, and that enzyme has two catalytic domains, and we are called HNH and ROVC. And the enzyme coats both strands of the DNA, and then repair of the DNA follows. And we humans employ CRISPR-Cas system by modifying those two catalytic domains of Cas nuclease, and we also modify guide RNA so that we could precisely target DNA sequence. And guide RNA is also called single-guided RNA in other papers. So after viral DNA, let's say viral DNA, it is cut, then virus wants to repair its DNA by either non-homologous end-joining or homology-directed repair. This one is said to be more precise, whereas this one is said to be more error-prone. So an example of non-homologous end-joining is Crispr interference. That includes, that means that to the target sequence, Cas9 nuclease binds and just silence the sequence, meaning that no transcription factors can bind to that DNA and transcribe RNA from DNA and then make protein. Also, DNA can be tried to be repaired by deletion or insertion so that the gene is inactivated. So I will focus on treatments by CRISPR-Cas system of those diseases. It's sickle cell disease, thalassemia, collagen mutation caused diseases, cancer, and also I will talk a bit how CRISPR-Cas system could be used to improve antimicrobial and antiviral applications. So these are the resources that I used for this presentation. So sickle cell disease is a prevalent disease that is mainly linked with hemoglobin, that the structure you can see there. It's composed of 2 beta chains and 2 alpha chains, and in each of the chain you see heme group that binds oxygen. So one hemoglobin molecule can bind 2 oxygen molecules and carry oxygen throughout your body. And this disease is a result of both hemoglobin beta chain genes being mutated, meaning that we have faulty beta chains of the hemoglobin. It's autosomal recessive trait, those which heredity is shown in this picture, and it's monogenic disease, meaning that it's quite easy to target, easy to treat with genetic engineering tools, because we can try changing one gene and then the disease should disappear. And nowadays, treatment is just trying to disassociate the fibers, because due to that mutation, as you know, red blood cells aggregate to each other and are not able to transport oxygen effectively. And genetic tests are being done before doing sport, so that if a person has this disease, then they are likely to get heart attacks. And this disease is caused by point mutation that results in an amino acid sequence of beta chain that has valine residues instead of glutamate. So this is how a red blood cell looks like when it's from a person that has a sickle cell disease. And I was searching for a diagram like that, and I found a lab who works with sickle cell disease in Cambridge. And so what are we searching in that disease is we are interested in altered potassium permeability and how it causes red blood cells to shrink. So that this shrinkage is related to potassium ion transport, as you see from those pumps. Yeah, and how that's potassium causes hemoglobin as polymerization and sickling. We also compare different genotypes of sickle cell disease. The 3 ones that I don't really know about, and we are not learned on them in lectures. The mechanism and prevention of phosphatidylserine exposure as a thrombotic and phagocytic event, and potassium chloride co-transport. So why does hemoglobin as aggregate? Why cannot it transport oxygen? So basically, here you see normal hemoglobin molecule composed of alpha and beta subunits, as I said, that has not bound oxygen to it. Then after oxygen is bound, we have that hydrophobic pocket of phenylalanine and leucine residues. And it's fine, and this hemoglobin can transport oxygen. But if you have hemoglobin S, meaning that mutated one, then you have that valine instead of glutamate, or at 6 amino acid residue. And what happens is that these triangles can perfectly fit into that hydrophobic pocket, and because these two amino acid pockets are hydrophobic, they attract each other, and we have deoxy as aggregation, and yeah, that's it. And the interesting thing that although hemoglobin is one protein, it has subunits that are expressed from different chromosomes. So alpha chains of hemoglobin are expressed for 11th chromosome, whereas beta hemoglobin chains are expressed for from 16th chromosome. And you may expect that we will have excessive amount of alpha hemoglobin chains because it is coded by 4 alleles on one chromosome, whereas beta hemoglobin chains only 2 alleles of the maternal and paternal chromosomes. So how do we go around that? If that would be the case, then excess of alpha chains and normal levels of beta chains, alpha chains would aggregate. This is alpha chains, sorry. But we have alpha hemoglobin stabilizing protein that is quite abundant in our blood, and this protein binds to alpha chain monomers and forms soluble complex, so that we go around this problem and we have normal hemoglobin because of that stabilizing protein. So hemoglobin is allosteric transporter. It transports oxygen very efficiently. Whereas, for example, myoglobin is a storing oxygen unit in heart and skeletal muscle cells. And the thing is that it binds oxygen so tightly that when this protein goes into the tissues, not much of oxygen is released, because oxygen is always bound to that myoglobin, and therefore tissues don't get much oxygen. And this is why hemoglobin is a much better oxygen transporter. And interesting thing is, from the myoglobin concentration in meat, you can get the color of meat. You can see why you have different colors of different kinds of meat. Now, what about fetal hemoglobin? So we talked adult hemoglobin has alpha and beta chains, whereas fetal hemoglobin has alpha and gamma chains. And this allows fetus to get oxygen efficiently from mother's blood. So how do we go around this? Like when we are babies, we have this kind of hemoglobin, and later we have hemoglobin composed of different subunits. So how does that happen? So actually, there is this BCL11A protein that represses gamma globin synthesis when we get older, and CRISPR-Cas system targets this repressor so that people with sickle cell anemia have their genes edited so that they start producing fetal hemoglobin, hemoglobin with gamma chains, and their oxygen delivery to tissues improves. So how was this found, that how this idea came that such CRISPR editing of hemoglobin gene could improve sickle cell disease symptoms? So it was found that single nucleotide polymorphisms of chromosome 2 for this locus was correlated. Polymorphisms were correlated with lower severity of both thalassemia and sickle cell disease. So these cells were taken, edited with CRISPR, at enhancer region of this protein, and patients that had that genetic editing had reduced expression of this protein in erythroid lineage cells and restored gamma globin synthesis. So their hemoglobin were synthesized in the form that is found in fetuses. Another disease, thalassemia. It is also a monogenic disease, and it can be either alpha thalassemia or beta thalassemia. If we are talking about alpha thalassemia, then no or little alpha chains are formed, and we have hemoglobin tetramers with beta chains only, and then hemoglobin does not show any cooperativity in oxygen binding, meaning that hemoglobin binds oxygen with very, very high affinity, as we saw example with myoglobin, and this leads to poor oxygen release to tissues and poor solubility of whole hemoglobin protein. And sometimes this kind of thalassemia can be fatal. The paper I found with CRISPR was about beta thalassemia, when we have no beta chains of hemoglobin, and we find precipitates inside immature red blood cells, and this leads to anemia. So what was done to treat beta thalassemia? We see here, oh yeah, so it's again absolutely the same what I explained, just a picture of that. So these are the exons of BCL11A protein, and so clinical trials with CRISPR-Cas9 system are very expensive. So only 2 patients were tried to be treated with CRISPR. So one patient had that thalassemia, transfusion-dependent thalassemia, and another patient had a sickle cell disease. And they both were gene editing for them performed the same at the same BCL11A gene, and these patients were followed for at least 1 year, and later 8 additional patients were also this kind of genetic editing was performed for 10 other patients then. And these are the clinical trial, like for these 2 clinical trials, when each had only 1 patient. And yeah, and infusion of Cas9, infusion of those autologous edited CDR4 plus cells that I wrote on the last slide, they're infused only once, and this required just monitoring of patients. So the results seem to be quite outstanding because both patients had early substantial and sustained increases in fetal hemoglobin levels with more than 99%. All of like normal pancellularity refers to like proteins or some cellular components at whole organism, like at organismal level. So during a 12-month period, and this indicates that the gene editing effect was durably maintained. However, everything is not so successful as it looks like, because investigators classified 2 adverse events as serious, was pneumonia in the presence of neutropenia, and also veno-occlusive liver disease with sinusoidal obstruction syndrome. And we began on study day 13 when it was resolved on day 28, when on day 39, another symptom was resolved. So these patients need very, very, very close monitoring because complications can result after I think off-target CRISPR-Cas system cleavages. So now maybe somebody wants to guess what is the most abundant protein in humans and mammals, because next slide is gonna be about it. Does somebody know that? No one listening?
We just don't know.
Really? The most abundant protein. Okay, I also didn't know that. So it's collagen. It's in our nails, hair, skin, and 4 stages. 4 steps are needed to make collagen. So first of all is procollagen synthesis. Second one is cleavage of that procollagen to make tropocollagen. When tropocollagen is assembled and we get collagen fiber, when collagen cross-links with lysine and we have a stronger fiber. This is how it looks like in a diagram. This is procollagen synthesis and secretion. So it's synthesized in the endoplasmic reticulum when we need vitamin C for hydroxylation of collagen protein, hydroxylation of proline and lysine residues. And if we don't have vitamin C, when we get disease scurvy, which results in abnormal skin, when we have release of collagen from endoplasmic reticulum to the cytosol, so that we have procollagen where that is glycosylated, when we have triple helix formation, and this is called procollagen. When it is secreted out of the cell to the extracellular space, and procollagen peptidase removes N and C terminal domains of the triple helix, so that we get to third stage, which is collagen assembly. And so lysyl oxidase cleaves tropocollagen, and collagen fibrils are assembled so that we get lysyl oxidase makes lysine residues linked to each other so that we have extensive fibrous collagen network that makes up our skin, hairs, nails. So now a bit about collagen disorders. Why is it a genetic disease? Because I'm not sure I can see that, but there are many types of collagens, and we are made of different genes, so that a mutation in any of the gene can cause abnormalities in all of these tissues, organs. So first, like second disorder after scurvy is osteogenesis imperfecta, and it is caused due to mutation of glycine to cysteine. So maybe you remember that collagen is composed of glycine residues that compose 30% of collagen, and around 63% of collagen is composed of proline residues. So glycine is a very important amino acid, and if you have mutation that cause absence of this amino acid, you have disrupted collagen helix. Another syndrome is Ehlers-Danlos Syndrome, and there are many types of them. This is just one type of the syndrome that is caused due to defective removal of propeptides, and this is because procollagen peptidase in stage 2 of collagen synthesis cannot perform its function, and this leads to increase in collagen in the skin and tendons, so we have too much collagen. So there are many Ehlers-Danlos types of the syndrome. So we just talked about defective procollagen peptidase. There is also another type of the syndrome when we have inhibited lysyl oxidase, and this is called lepharism. So beta-aminopropionitrile is in the food of cows or cattle, something like that. And sometimes if the food is contaminated, and then what these animals get is that we cannot cross-link collagen fibrils, so you have kind of separate triple helices of collagen that are not cross-linked, and this leads to abnormalities of joints, bones, fragile collagen, and too large vessels. Yes. Oh, so this was inhibition of lysyl oxidase. And here I actually wanted to talk about another Ehlers-Danlos syndrome, which results in hypermobile joints, hyperextensibility of the skin. And it is caused by gene mutation that results in no lysyl oxidase present. Then how do we treat all of these collagen gene, all of these syndromes caused by collagen gene mutation? So one example that I found is for treatment of type 7 collagen gene mutation, and this is a recessive dystrophic epidermolysis bullosa, the name of the disease, right? And so what do we have there? We modify that collagen gene with CRISPR-Cas system so first we take skin from a patient, right? Then we modify genes in that skin. Then we add Yamanka factors. Yamanka, the scientist I talked with you about last journal club. Then we get induced pluripotent stem cells. And when we create such an environment in the petri dish, that we get get correct keratinocytes with a correct genome, and so that we regain functional type 7 collagen protein, and we regain complete attachment of epidermis to the dermis, and thus that patient, after we re-inject those cells to the patient, that patient should not have severe blistering, fibrosis, predisposition to a type of cancer, type of skin cancer. And this disease is quite This disease also results in corneal and esophageal lesions and decrease in life expectancy, so that these collagen-related diseases are quite important because collagen is the most important protein in our bodies. Now a bit about cancer. Now let us pass on to cancer and how CRISPR can be employed better. So we can target with CRISPR tumor suppressor genes such as PTEN and TP553. And by doing that, we can understand more what genes contribute to cancer, either individually or combined. So tumor suppressor genes saves us from cancer. And if we knock out those genes, for example, with CRISPR, we just remove those genes, we can see if cancer symptom, if cancer becomes less metastatic. When CRISPR-based screens have enabled identification of essential genes and drug targets, and CRISPR system allowed us to do comprehensive analysis of cancer mutation profiles, and may one day highlight mutated proteins that could be targeted for therapeutic intervention. For example, CRISPR system allows us to repair a nonsense mutation in the tumor suppressor ASXL1 in a mouse model of myeloid leukemia. And this kind of gene editing has been shown to extend lifespan. And about antimicrobial applications. The interesting thing is that we can make microbes target themselves with CRISPR-Cas systems, and this would be much better than antibiotics because when we use them, our good bacteria, microbiota, are also destroyed. And the thing that is only needed for these cell-targeting CRISPR-Cas systems to be employed is we need robust delivery options to humans and then proceed to clinical trials. So regarding viruses and their removal, actually there are CRISPR-based therapies that are being tested, for example, to destroy HIV, herpes, papillomavirus, hepatitis B virus. And regarding HIV therapy, editing the host or targeting the virus have been tried, and co-distribution of genes encoding the HIV cell surface receptor proteins, chemokine motive receptor 4 and chemokine receptor 5 shows promise in preventing the HIV entry into our T cells. And also there are some genes in pigs that if we use a tissue of a pig and want to, for example, transplant to humans, when we also transplant the gene that encodes the virus of that from that pig's genome. So to prevent that risk of spreading of that virus, we could use CRISPR to target that one gene in pig's genome to remove the probability of us being infected with the virus. And a bit beyond treatment of diseases, I saw somebody from our society very interested in biofuels. So we work primarily by taking up CO2 from the environment and then when they are being destroyed, they produce energy. So so greens by themselves, we have chlorophyll, so they rubisco enzyme, we take CO2, we integrate that, that's fine. But the problem with algae before were that when you burn them, they do not produce much energy, and this was because they didn't contain much fat, and thus biodiesel from there was really impractical. So what CRISPR did, CRISPR was used to create strains of algae that produce twice as much fat, and fats are molecules that store a lot of energy, as you know. And to produce fats, we need more CO2 molecules, so more CO2 is taken up from the environment, and we have practical biofuel. So synthetic genomics works now with ExxonMobil to meet the target of producing 10,000 barrels of algae biofuel per day by 2025. Another short application is of CRISPR is eradicating pests. So last year, researchers at Imperial College London proved that CRISPR could be used to eradicate mosquito that carries malaria parasite, Plasmodium. So they introduced a gene that when their two parents carry that gene, then those insects stop laying eggs, and in trial with cage mosquitoes, the population was wiped out as soon as 7 generations after the gene drive was introduced. And the last application I wanted to talk about is, I think, the most famous one. It's the first thing that comes to my mind when you talk about CRISPR-Cas system. So it's myostatin gene. So this gene is negative regulator of muscle mass, meaning that people who have a lot of myostatin protein, they will have reduced muscle mass. So if we want to have a horse that runs very fast, we want that horse to be muscular. So we knock out that myostatin gene, and we get double muscle in horse, or in mice, goats, pigs, and cattle. And recently, it was done in rabbits. So we simply co-inject Cas9 nuclease mRNA and single-guided RNA into zygotes, and this is the result we get. So the Argentinian company is editing the genome of race horses to make breeds that are faster, stronger, and better jumpers. They have produced healthy embryos, and we expect the first horses to be born this year. Maybe this year, like I'm citing this, so maybe this year was already a few years ago. And the last point I wanted to make is about what is more promising is this epigenetic modifications or gene editing. So actually, they are not so separate fields. They are quite interrelated, because epigenetic silencing or reactivation of a mutant allele could be a potential therapeutic approach for diseases such as Rett syndrome and Huntington's disease. So we, for example, another nuclease, not Cas9 system, but other gene editing tool, and transcription activator like effector nuclease When fused with transactivators, they are very feasible methods for epigenome editing. However, these two nucleases are hard to synthesize, and therefore we are labor consuming and costly. So what can we do? We can use Cas9 system that is those synthesis is cost effective and much more easy to manipulate. So we just redesign the programmable guide RNA and synthesize new expression cassette. And when epigenome editing begins to thrive because of CRISPR-Cas system. And here I found dead or deactivated Cas9 that has no nuclease activity but functions as a DNA binding domain. So epi effector Cas9 protein with a transcription repressor or activator are shown here. So here we see that deactivated Cas9, and here you see the epi effector. And so we are fused, and when we bind to the red PAM sequence, that is just after the target sequence we would cleave if we have a normal Cas9 protein. So after this binds, DNA methyltransferase adds methyl groups on DNA, and we have decreased the gene expression. Oppositely, if epi effector is TET, a DNA demethylase, when we result in demethylation of DNA and increased gene expression. The highest methylation rate was estimated to be 50%. That is, you see, not 100%, but it's still promising. And this increased gene expression can be long-term. So not only DNA epigenome editing can be performed with CRISPR-Cas system, but also histone modification. So again, we have that epi effector and Cas9 system that binds to histones and results in increased gene expression. And also we similarly can get decreased gene expression by modifying histones. And this was an interesting thing that Cas9 protein can be coupled with fluorescent dyes, and we can image live cells so that electron microscope and other many microscopes allows us to image only dead cells, whereas here we can see have a huge advantage of monitoring, for example, telomere length dynamically. So we can deliver guide RNA into the cells at different stages, and we would see observable fluorescent signals, to some extent reflecting the length and conformational structure of specific genomic loci. So thank you everyone for your attention, and as it is the last journal club with me this year, so I thank you for this privileged position, for the very organized president, for exceptional audience, for brilliant graphics, for our events that were wonderful, and thank you for all the speakers that you invited. And I'm waiting forward to your events next year. So my friend is gonna be the head of a journal club. So I think I will know everything what you are guys doing there, and I wish you very good luck there. And I wanted to share my views, what can be improved on this position, or just some ideas for committee members maybe. So my idea was that if PhD students or if somebody certified, I don't know, would run journal clubs, we could give certificates for attending or our events or something like that. Then so this would give motivation maybe. Then I think that more feedback from the audience would be helpful for the person organizing journal clubs. So that this could be Google Docs for maybe at least for, I know, first 2 or 3 journal clubs about content duration or conciseness. But also I thought that we could actually a more efficient way would be if a few people would say their opinion, their just comments after the presentation, and we actually could be a different role in the committee. For example, we could be like supervisor would not be a role, but we could be mentors, so that one could say English mistakes and how to improve. Then another could concentrate more on content and reliability, because some of my, for example, sources, they're not from like Nature or from Science Direct. Some of them were just from websites. And then yeah, so shortly about that. So I think that okay, so about speakers. So I think that this is, since I am not an expert, I think instead of speaker in maybe in Instagram post, maybe it should be better said presentation by, but it's just a detail. Then if the feedback would be given, after representation, when this position would become like more prestigious, I think, because it would allow a person to improve more. It wouldn't be just for that person just to reading more about the topics, and also that person would gain more skills due to the feedback, and it wouldn't be just an experience of presenting. And the head of the journal club, I think, would be better changed to organizer of the journal club. And so that person would, I think, more applications maybe would be to this position because that person would get feedback. It would be like a mentorship where because if after every journal club, for example, the same people that could call themselves mentors even maybe, they could give that person valuable feedback, and it would be like academic development scheme. So I took this from UCL Investment Society, and we really have mentors. So I thought, why couldn't we have like free mentors, for example, for that one organizer of a journal club? And the thing is that then we would probably need like third year or at least second year students that are into genetics, are studying something related to genetics or biochemistry or something like that, and could really advise. So I think that this would be a great add-on to the CV, a better add-on on the CV of the organizer of the journal club, and also to these mentors. So thank you very much. And do you want to ask something or give feedback, I'm happy to hear anything. And these were just my views. So if you don't like them, you can just forget them.
Yeah, I mean, I thought that would be really useful. Would you be able to send me the last few slides so that I can work on that with you, Han, for next year? I agreed. I do think they're useful things to do, and I think they'll improve the journal club for next year. Thank you.
Great. Sure, I will.
So yeah, I don't have any questions, but I just wanted to thank you once again for the incredible job you've done by being the head of the journal club this year. Like your presentations are always so thorough and like detailed, and I just think you've done an exceptional work, given the amount of work we need to do for uni, the quality of the presentations, it's just mind blowing. So thank you so much, and thank you for engaging us and widening the knowledge in the genetics field. It's amazing.
Yeah, thank you very much for founding this society. It's very nice to contribute to valuable things, to meaningful ideas.
Please come back to Genetic Society next year. I can't imagine Genetic Society without you.
That's fine. I will be there with Tracy.
Yeah. We still have a couple of positions free. So like if you would want to apply there.
No, thank you. I have other plans.
Okay.
But so I think I could mentor Tracy because she is gonna be the head of a journal. Okay, I don't like that, head of a journal club. It sounds like the head of a kindergarten, and when it's just organizing journal clubs. So I think when she will do presentations, I will help her. Maybe I don't know. It's personal, but it's actually mentorship. If there would be like a jury, maybe, I don't know, like three people, if for example president, vice president, and another person would always say after presentation, like comments like for 1 minute or 2 minutes, but not only just like only pluses of a presentation, but also pros and cons. It would be very helpful for her and also for those who comment.
I was thinking maybe we should do this, like we could try doing this with mentorship scheme, and we can also try to do it like an anonymous vote, like what people liked, what like point, like qualities of presentations were good, what drawbacks. So, and make it anonymous so people don't feel like of course it could lead to excessive critique, but I don't think that we would have like hard really critical people next year. I
No,
think
constructive feedback is always okay, and if it would not be anonymous, I think it would be fine.
okay. And also, would you be okay with creating a Google Drive link to all of the slides you had presented this year? Because
Really? Well, yes, I think so.
okay.
Yeah, sure.
And thank you once
Sure.
again. It was incredible work.
And even with English, I thought, guys, everyone who is in the audience, even you all saw my English, like I don't know, gaps or something, and any of you, I think you could have created like of, for yourself, an add-on on CV, for example, if you write down my mistakes when give me back to me when you say it's attention to detail, it's English, something like that. So I think that mm, like for the next year, if somebody would like to do this, I think that like feedback on English would also be necessary, so you can expand the range of positions in your committee.
Yeah.
So thank you very much and have a nice evening.
Have a nice evening as well. Thank you.
Thank you. Goodbye.
Thank you.
Bye.